function of the retina is a vision. Vision is nothing but perception of objects based on the light that they emit or reflect. So normally human can able to detect a visible narrow range of an electromagnetic radiations ranging between 400 nanometer to 700 nanometer. So the function of this vision starts from the photoreceptors. So as I already told, there are two type of photoreceptors. One is a rod, another one is a cone. Rods helps in night vision, whereas the cones helps in day vision and the color vision. So again, when it comes to cone, we have three type of cone: L cone, S cone and the M cone. Each absorbs different wavelength of the light depending upon their pigments. So all these photoreceptors, they able to perceive the light because of the presence of the visual pigment. In case of rod, it is rhodopsin, and in case of cones, it is rhodopsin. So when you dissect these pigments, we actually get opsin and the retinal. So this retinal is common for all the pigments, whereas the opsin is different for each of the photoreceptors. So when these two combines, helps in the absorbing different wavelength of the light. So mainly the opsin is responsible for the absorption. So now what happens in the dark? That there is a thing called dark current is created. So in the outer part of the photoreceptor, an element called cyclic GMP is generated. So this cyclic GMP will help in opening the sodium gated channel, where there will be influx of the sodium, and because of this influx there will be around minus 40 millivolt local potential is created. So when this is created, a neurotransmitter called glutamate is released in the photoreceptor bipolar junction. So to keep this dark current active in the retinal, we have two components. One is a cis retinal, another one is a trans retinal. The cis retinal will be present in the dark and it is an inactive opsin. So this inactive opsin will keep the cyclic GMP activated. So again I will repeat, in the dark the cis will keep the cyclic GMP activated. The cyclic GMP open the sodium gated channel. There will be influx of the sodium. Because of this, a neurotransmitter called glutamate is released on the photoreceptor bipolar junction. Now light enters the eye, where the cis retinal is converted into trans retinal. This trans retinal stops the cyclic GMP, so then the sodium gated channel will be closed, and then influx of the sodium will be stopped, and also the release of the neurotransmitter glutamate will be stopped. I will repeat it. Light enters the eye. From the cis retinal, trans retinal is formed. The trans retinal will pushes this cyclic GMP. Sodium gated channel will be closed. Influx of the sodium will be stopped, and so then the release of the glutamate also will be stopped. Now the bipolar understands that it is receiving a light, so it generates the action potential. It from the ganglion cell, it reaches the optic nerve to the brain, and the image is perceived. So actually the same process happens in both the rods and the cones. But what happens in the rods? On average, each GCL receives signals from hundreds of the rods. So the action potential generated by the rods will be more. So they have a high sensitivity. That's why they are able to detect an image in the dark. But these are activated only by a dim light. So their signal produced by them will be very weak. But together it forms a strong signal. That's why in the dark you are able to identify a person, but a sharp features you will not able to identify. So overall the rods will have high sensitivity, but the resolution will be low. But whereas the cones, each of the cone will connect to a single ganglion cell, so their resolution will be more. So when you see a person, you will be able to understand, you will be able to identify his nose. Even a small mole on the face you will be able to detect. But on an average, each GCL receives signals from the single cones, so their action potential is not enough. That's why the low sensitivity of the cones. This is the reason why you are not able to detect the color in the dark. Okay, so now there are other cells like horizontal cells and amacrine cells. So what it does is, it receives signals from the different part of the object. For example, if you take a car, the different light intensity will reflect from the front or from the roof or from the back. So each of this will be identified by this horizontal cells, amacrine cells, and they helps in understanding the contrast and the edges of an object. In short, you can say helps in spatial recognition. There are some ganglion cells where it receives direct light, as I already told, photosensitive light. So have you ever wondered, when you put a torchlight, how the pupil constricts or dilates? So it is because of these photosensitive ganglion cells that decides what is the amount of, that is intensity of light that should reach the pupillary pathway. That's why in case of any ganglion cell damage or any pathology involving the retina, the ganglion cell absorption of this light will be reduced. So again the light transmission will be reduced, so that's where we end up in an RAPD or a sluggish reacting pupil. Then we have few other photosensitive ganglion cells where it helps in the sleep wake cycle. So the overall, the first order neuron formed in the bipolar cells, second order neuron in the ganglion cells, the action potential is transmitted to the optic nerve. So optic nerve of both the eye converge, forms the optic chiasma, then the optic tract, then it reaches the lateral geniculate body where it forms a third order neuron. So from the lateral geniculate body it forms the optic radiation and then the visual cortex. So we all know that nasal fibers crosses each other, whereas the temporal fibers do not cross. So this forms a visual pathway. So some of the fibers, after crossing the optic chiasma optic tract, they reaches the midbrain. Okay, so this constitutes the pupillary pathway. So that's why if you get a lesion, that is if you get a field defect with the normal pupillary reaction, it gives a clue that the lesion must be somewhere beyond the optic tract. So the left side of the visual field will be perceived by the right side of the brain, and the right side of the visual field perceived by the left side of the brain. So to conclude, to master the surgery and pathology, first understand the anatomy and physiology, know the structures, grasp the function, then heal with the precision. So these are the common questions where they ask in our basic papers, that is anatomy, biochem and physiology papers. So this slide presentation will be, I think, helpful for all the exam going batch. Thank you.